Hello, everyone, and welcome to this third open mic session here, live from Novartis campus. My name is Rahel. I'm the community manager at Switzerland Innovation Park, Basel area, site Novartis campus, and I'm going to lead you through this session today. And I'm happy to hand over. Thank you very much. The stage is yours.
Thanks for this introduction. One comment, which I also posted here. So we're actually in the same building, and we're really happy to be hosted here, but not in the same room. So we decided to do this as a full online event to be joined with everyone online only. So yeah, I'm going to start my presentation as well, and I'm really happy to present what we do at Scailyte, what type of partnerships we're looking for, and I'm happy to engage with the community here. So yeah, now. Milan, if you can share my presentation. Yes, great, thanks. Yeah, so as Rahel already mentioned, I'm the CEO and co-founder of Scailyte. We're a Swiss-based startup. Our headquarters is here in Basel, in the Basel area innovation park. We also have an office in Boston at the Swiss Next there. And we started as a spin-off from the ETH Zurich 4 years ago. So in 2017, we focused on developing a single-cell analysis software, and I'm going today to explain how this works and how it could accelerate cell and gene therapy R&D. Right, but before I move to more the detailed and technical part, I want to start with a story. And here on this picture, you see Emily. Some of you may have heard this name, Emily Whitehead. And this is a picture of her in 2010, when she was at the age of 5. And that's the picture just a few months after she's been diagnosed with leukemia, and in fact, one of the most common leukemias in children, ALL. And this also marks the beginning of her patient journey. So when she was diagnosed with this disease, the doctor said, yeah, the chances to respond to the standard treatments and to be cured are pretty high. So 85% of children actually respond to the standard treatment. But unfortunately, Emily was not one of these patients, and she didn't respond to the standard treatments. So her patient journey went on for 2 years, and it relapsed twice. And in 2012, when it relapsed for the second time, the doctors almost gave up, and they recommended actually palliative care. But luckily, her parents didn't give up, and they went on and looked for solutions. And then they met Carol June, a scientist and a pioneer in the CAR T-cell field. And then in 2012, Emily became the first child to receive CAR T-cell therapy. And against all odds, actually, this worked, and she was a complete responder. And yeah, now she's 9 years later, alive and cancer-free. And yeah, what CAR T-cell therapies are is what they did with Emily is they took T-cells from her, they modified them in the lab, and then infused them back in the body to fight the cancer. And they actually did this job which no other treatment that they tried on her could do. And this shows the potency of cell therapies. But there are also challenges with this. So in 2017, the CAR T-cell therapy that was administered to Emily was also approved by the FDA, Kymriah. And now today we have 5 cell therapies on the market approved. And actually more than 500 clinical trials for actively developed CAR T-cell therapies. So you see that this is an emerging field, and in the future we'll see many more cell and gene therapies coming to the market. But one of the challenges is, for example, in the Kymriah clinical trial, only 39% of the patients had complete response. So there are still many patients that do not respond completely or at all to this therapy. And this is an issue because this, as I explained, it's a last resort therapy. So if the patients do not respond to this, unfortunately, they have no other options, and they will likely die within a short amount of time. And the other issue is the cost of these treatments. So for, again, Kymriah, for example, the cost of the treatment itself is around $400,000 US, but most patients also experience toxicity effects, and these costs to manage the toxicity can go up to $600,000 per patient, which makes the total cost per patient to go up to $1 million. And this is a huge burden for the patients and also for the healthcare system, and it could be actually managed by reducing the toxicity of the treatments. And this is also what we're now aiming to do at Scailyte with the technologies that we developed. So what we do is we leverage 2 cutting-edge technologies, one of them being the single-cell technologies, and the other one AI, so artificial intelligence algorithms. And what are single-cell technologies? So this is a new method or a new set of technologies that allows molecular profiling of individual cells at very high throughput. And just to be more specific, so if you take a blood sample from a patient and you perform single-cell RNA sequencing on this sample, you would profile tens of thousands of individual cells, and ultimately you would get more than 100 million data points from just an individual sample. And like this, you have really deep insight and deep resolution to understand the biology of a patient and what happens in the sample. But this comes also with the challenge. So these hundreds of millions of data points just from the single sample, this is an overly complex data set, also a very large one. And the conventional and standard data analytics tools that are used, they just struggle or can't cope with this complexity. And yeah, that's what we've been doing at Scailyte in the last 4 years. So we developed a data analytics pipeline that leverages deep learning algorithms and actually best-in-class deep learning algorithms for this task that bring the insight from single-cell data and turn it into clinical applications. And the clinical applications in the context of cell and gene therapy products could be in two aspects. So one on the level of the patients, and specifically to stratify patients by identifying gene signatures or cell types that could be indicative of the patients who have the highest response. And then on the second level, or the second aspect, would be on the cell product itself. So I mentioned with CAR T cell therapies, you actually take out the cells, you modify them in the lab, and then you bring them back into the patient. And this is the cell product that you actually use as the treatment. And these cell products also could be analyzed using single cell technologies and our AI. And like this, you would be able to identify the most potent cells before you administer them. And to be even more specific, I show here one example. So here we used a public data set from a paper that was published last year in Nature Medicine. And what the data set comprises is actually single-cell RNA sequencing data from 23 CAR T cell products. Again, so here we're looking at the cell product, not at sample from the patient. And these cell products were administered to patients with B-cell lymphoma. And out of the 23 patients that received this treatment, 9 had a complete response, so CR, and 14 had partial response or progressive disease. And now we set the task to our software to analyze the single cell data and look for a signature or a pattern in the data that could predict the response based on what is the composition of the cell product, in terms of molecular profiles. And the way how we do it is we split the data set into 70% and 30% of the samples. We take 70% of the samples to train an AI network, so basically find the pattern and train the AI model. And then once we do it, we use the remaining 30% as a validation cohort. And there we apply the trained network, and we try to predict the responses. And when you do this, you can come to the next step and then have two outputs or two readouts. So the first one is we measure the accuracy of the prediction. So based on the pattern that we learned from the initial data set, what is the accuracy of prediction? And in this case, we achieved 87.5% accuracy of predicting the response of the patient based on a signal from the cell product. And this is already quite impressive, but even more so, we can dive deeper and also understand the biology or what is explaining this prediction. And there we discovered also relevant cell types within the cell product and inflammatory markers, which expression actually explains the association with complete response or no response. And this is one example where single cell analysis or really high parameter molecular profiling of individual cells, again, either on the cell product level or from the patients, combined with advanced data analytics, can bring significant improvements and also applications that could be used on a routine basis. Because now this model that we trained, again, it could be reused also to predict the response rate of other patients or unknown cell products before you put them in the patient. So that's, let's say, a prototype application, which if we have more data and the right partners, we could really drive forward. And when we say we have a best-in-class algorithm, many actually AI companies in all different industries claim this, but we also have the evidence to prove or to back our words. And single cell data, I already explained, it's a very new type of biomedical data, a very complex one. So the algorithms that work, so there are many deep learning algorithms, for example, for image analysis or analysis of other types of data. And when you try to use them on single cell data, they don't work just because of the different format and complexity of the data. So in the single cell space, there is a lot of now research being done, many new algorithms are being developed, but they're all tailored to analyze single cell data. So what we did here, we took a very massive data set of single cell data, And we benchmarked ScaiVision, our software, against all the most common algorithms that are used in the single cell space. And here the test was actually similar to what I just explained. So you take 70% of the data and you train a model, and then you use the remaining 30% to test the accuracy of the model. And the readout, this AUC or area under the curve, in the case of perfect performance, then you get a value of 1.0. In the case of just random prediction, you get 0.5. And yeah, we were very happy to see that our software performs really amazingly on this very massive data set with an AUC of 0.95, while no other algorithm could achieve this. And you see some of them are even close to random prediction. So yeah, this gives us the confidence to develop further our algorithms and pipelines, and again, apply them in different clinical settings. And here also to stress again where on the workflow of cell therapies we could make an influence. So here we could definitely improve the donor selection or also later the patient selection by analyzing samples from the patients and predicting the response or how the donor material could work for producing the cell product. Then at the end of the manufacturing product, when the cell products, before they get delivered to the patient, there also you could apply single cell analytics and biomarkers derived from single cell analytics to improve the quality assessment and again to find the most potent cell products before they go in the patient. And then finally, once you infuse the patient with a product, you need to monitor again how they develop, whether they develop toxicity, and whether the product is still working. And that's again a point where we could address with our approach. And now to finish, coming to the beginning with the story of Emily. So this is a picture of her from beginning of this year. And I mentioned already, so she really had a complete response and has been cancer-free for 9 years. And this foundation that she and her family started, the Emily Whitehead Foundation, it really promotes research, because what saved Emily was this amazing breakthrough research that Carl June has been doing in his lab. Which obviously is possible only with significant investment. So here also a call to action to anyone who is in this field, really to support foundations like the Emily Whitehead Foundation, and also to actively think about and promote cancer research. And yeah, that's also what Scailyte is doing. And we're now really in the field where we see our technology that we developed can really influence and accelerate the research and the discoveries in the cell and gene therapy space. And we're now actively looking for partners from this space to advance together the cell and gene therapy products. So yeah, if you're interested to talk to us, I'm first happy to address some questions now, but also you can contact us at contact@scailyte.com. Also, you can find me on LinkedIn, and I'm really happy to get in touch with you. Thanks. Rahel, back to you.
Peter. Thank you for this very insightful presentation. Yes, I'd like to open up now the round for questions. I'm not sure if there are any. Maybe we can start off with a question from my side. So I'd like to know, actually, since you've been on the way a couple of years now, how is it going? How much money have you raised so far? And are you currently looking for new investors?
Mm-hmm. Yeah, thanks for the question, Rahel. So we've been in the startup business for 4 years, and we did a few fundraising rounds so far. So until now, we raised 7.5 million Swiss francs. And actually, just now in the next couple of weeks, we're going to close our next fundraising round. Again, with the aim also to support the development of applications in the cell and gene therapy space. That would be one focus area. And we're also active in women's health in a disease called endometriosis. So we'll use some of the funds also in this direction. And actually, we're also still looking for new investors, and really investors who are passionate about what we do, investors who are also from this space, from the healthcare space, or in particular from the cell and gene therapy space. And this could be private people or venture funds. And now when we close this round, we still have a 6-month period where we could close an additional investment. So if someone is interested in investing in Scailyte now, please also reach out. And in the next 6 months, we can still include new investors in the current fundraising.
Thank you very much. Now a few questions actually came in. So there is one asking, can the algorithm also be used to establish disease severity indicative biomarkers from patient cells?
Disease severity indicative, yes, it can. I'm just thinking whether we already did such a use case to give the example. Disease severity. So it could predict the disease progression. Maybe that's where the question points to. So if we have the right endpoints, so ultimately how our algorithm works is it takes the single cell data and it looks for a pattern that associates with a clinical endpoint. And if this clinical endpoint would be the disease progression, so whether a patient would have severe progression versus mild or kind of staying stable, then we could train a model which could predict from an earlier time point how the patient would develop. So that definitely is possible with our approach.
Thank you very much. One more question from Ashley is if you can elaborate on your single cell sequencing technology. Is it similar to 10x genomics?
Yeah, so this I didn't elaborate in the presentation, but we are actually using commercially available platforms to generate the single cell data. What is proprietary to Scailyte is the data analysis platform. So most of the single cell data that we analyze is actually from 10x genomics platforms, but we also look into CyTOF data, mass cytometry, and other types. And again, the algorithm that we have can look at any type of single cell data. But yeah, just nowadays, the predominant technology being used is 10x genomics.
Perfect. So Christoph is asking, actually, he says, amazing performance from only 23 data sets. Where do you expect the plateau if including more data?
Yeah, that's also a very good question on the data set number or the sample number. So actually 23 is almost the bare minimum with which we can start. Anything lower than this in terms of sample number would not produce a meaningful result. And ideally, you want to have something in the range of, let's say, 60 to 100. So we had also some projects where we had data sets of 60 patients. And yeah, there you get also a more robust signal. So that's, I would say the plateau probably would be reached somewhere around 100 patients for discovery. And optimal number is 50 to 60, I would say. It depends also how many conditions you're looking at and whether it's simple response versus no response, or something more complex like longitudinal studies. No.
Thanks, and I think Christoph has added the second question and asking, have you tried to include additional clinical or genetic markers in the model training?
Yes, we did this as well. Again, the core data that we use is single cell data, but then we complement this with other data types. And for example, in one project, we also include cytokine measurements, which obviously are not on the single cell level, but on the sample level. We also include a lot of patient data like age, other diseases. So we can build models that are more complex and go beyond just the single cell data.
Great. Then maybe something I'm also curious in, since in the last couple of months, you mentioned a number of new partnerships that you've entered. So what are the kind of partners you're looking for, and how do you normally collaborate with partners?
So at the moment, we're focusing on collaborating with companies that are doing active clinical research or develop drug discovery and development, where we could complement their efforts with our capabilities in single cell analysis and biomarker discovery, more like in service partnerships, and again, particularly with a focus on cell and gene therapy companies. So that's why we're now really actively looking for such contacts. So if any company is developing new cell and gene therapy products, we're really happy to get into discussion and see how the two technologies can come together and ultimately advance the products.
Great. I see two more questions. One is about, what about taking more care on donor selection? Would it be possible to store the product so that it can be sold later on, or is that ad hoc production?
So this maybe I would refer to my colleague Diana, and also maybe to introduce her now into the panel. So Diana is one of our biomedical scientists and a specialist in immunology, also driving a lot of the initiatives that we have in cell and gene therapies. So yeah, Diana, maybe you can jump in here.
Yeah, thank you, Peter. I'm not sure if I understand the question correctly, but the general idea is that the characterization of the final product and this information can be used in many different approaches. One can select the most potent cells that would be most effective in patients. However, this information can also be used in order to improve manufacturing protocols and generate more successful therapies in general for the next generations of CAR-T productions. So how this information is used in the end depends on the client themselves.
Thank you, Diana, for joining us on the stage. One more question appeared. It says, looks like it is the proper cell types that guarantee successful response. Could the selection of proper cell types will improve treatment?
Yes, I maybe also here, Diana, you can jump in, but my kind of high-level answer would be yes, the proper cell types, but also it goes beyond this, because if you once you understand also the signaling that goes along these cell types, you could also find other markers that are not on the cell level, again, like cytokines or other signaling molecules. But yeah, Diana, maybe you can also explain this better.
Yes, I'm also aware of the work of Marco Ruela, who presented it last week as well, again, 2 weeks ago. There are intrinsic mechanisms that can compromise the activity of these cells, which can be interfered with once we know which pathways would be compromised related to therapy response. In addition to that, there are cell types that correlate with toxicity, and selecting the right cell types that correlate and help effectiveness of the therapy and reduce toxicity is actually even the bigger advantage of characterizing the products in such detail.
Great. There was, I think, a response also coming in, or a suggestion. So maybe Diana, Peter, you have a look at that later on. Are there any further questions in the audience that we could address? Otherwise, I would, open up the networking sessions, the one-to-one sessions for those who would like to stay with us, and thank Peter and Diana for the very interesting presentation on Scailyte and their activities. Diana, for joining us during the Q&A session.
Yeah,
we will send. Yeah, thank you. We will send out the presentations and a survey link after the event to all of the participants, so you also receive my presentation. Can have another look at that. But yeah, then I will say, thank you very much for your attention. This was the third open mic and the last one for this year. We will be back next year with different formats. If you want to connect with us, with Scailyte, please feel free to reach out. If you're on the campus, make sure to stop by. We are in building 500. So make sure to visit us. And yeah, I wish all of you a very nice afternoon ahead and a great week. And thank you very much for joining us today.
Thank you.
Thank you, and bye-bye.